Well, thank you for DEBRA International for inviting me, and it's so nice to be here with all of my friends and colleagues, and I just feel such a sense of togetherness with all of you. We're all together trying to fight EB together, and it's such an important goal. You about dystrophic EB, it's a fairly terrible disease. It's characterized, the most severe forms, by erosions and fibrosis, mitten hand scarring of the hands. I'll just go to the next slide, please. And then also you can have esophageal strictures, squamous cell carcinoma, and so these are all devastating consequences. It's, can I have the next slide, please? And it's frustrating just for physicians and families and patients because up until now, we weren't able to actually reverse the blistering process. We could provide good wound care, and the wound care has gotten better over the years. We could treat infections, optimize nutrition, obviously, because these patients are walking burn victims, and they just have a very high metabolic requirements. Treat their anemia, and then treat their squamous cell carcinomas when they arise. But we really haven't had a chance to do anything to reverse this process. Can I have the next slide, please? So with collagens, with RDEB, the defect in this disease is gene coding for collagen 7. So the lack of collagen 7 in dystrophic EB skin is what causes this disease. We know collagen is a trimeric protein that forms a triple helix, and then it forms dimers, and then it polymerizes into anchoring fibrils, which are then the structures that hold the skin together, the basement membrane on top of the papillary dermis. And that's what we're trying to correct here. Can I have the next slide, please? So our first attempt at doing gene therapy was an ex vivo approach, where we took patient cells and then we put the collagen 7 gene in the patient cells in the laboratory, and then we took the cells that were expressing the corrected collagen 7 gene of the patient, arranged them into sheets, and then grafted them back onto the patient. The next slide, please. And this is what it looks like. We were able to show improved wound healing, collagen 7 expression you can see in the center panel, and you can see anchoring fibrils in the lower panel here. And so this worked. It varied from patient to patient, and it's certainly not a one and done type of a process. It worked well, and it's a durable therapy. It's a very safe therapy, but it gradually declines over several years. And so it was clear that it wasn't enough to permanently correct the disease. Can I have the next slide? So what I just explained to you was what we call ex vivo gene therapy, where we take a biopsy from the patient, we grow the cells up, we put the correct gene back in. This is using an integrating virus, which inserts randomly, and you could have a very small theoretical risk of actually inducing a cancer. And then we expand the corrected cells, arrange them into sheets, and then transport back to the clinical site. And this is a lot of tight logistics. When you're transporting these cells back and forth, that you can't do it frozen. They have to be alive cells, and there's only a very short window of time when you can get it back to the operating room, and then under general anesthesia, you place these grafts. So this is a technique that requires a lot of specialized expertise, like a pediatric anesthesiologist who's familiar with working with EB patients, because these patients have very fragile airways. But despite all these challenges, we were able to show some success. In contrast, there's another approach to gene therapy. Can we just go back one second? Called in vivo skin correction, where we just take the drug off the shelf, ship it out to the clinical site, and then put it on the skin. So much simpler. The next slide. And so I'm going to give you an example of in vivo gene therapy that I've worked with for the last several years. It's a product called BVAC, Baramagene Garpurpavec, and it is an HSV1-based gene therapy vector. And the gene therapy, this vector has a really great properties in many different aspects that I'll talk about. One of them is it's got a very high capacity. You can actually fit 2 of the very large collagen 7 genes into this vector with room to spare. And so it also enters into the nucleus, and it's episomal, so it doesn't insert into the nucleus, so you don't have the risk of cancer formation. Have the next slide, please. And one of the problems with in vivo gene therapy to date is this problem with immune reactions. Like our immune systems, the most important function of our immune systems is to fight foreign viruses, right? And so, and it does it quite well. And so up till now, all of the in vivo gene therapies that we've been using have created lots of inflammation. There was a patient who died after getting an adenovirus gene therapy years ago, which kind of set the field of in vivo gene therapy back. And then we developed newer viruses, such as adeno-associated virus, or AAV, which is probably the most commonly used in vivo gene therapy now. But still, there's still inflammation, especially when you re-administer the agent after the immune system had a chance to develop immune reactions against it. Have the next slide. But one of the nice things about HSV1 is that it has the ability to evade the immune system. So we all know that herpes can lay dormant in the skin only for months or years, only to reemerge later, which basically proves that the immune system can't clear it. It's not for lack of trying that we still don't have an HSV1 a vaccine, and that's because it's so good at evading the immune system. There's multiple mechanisms that people have described. This is probably one of the more important ones, where one of the viral gene products of HSV1 actually can bind to an antigen processing protein and prevent antigen processing, which is a key early step in the immune response against viruses. So it's not a great thing to have a disease where your immune system can't clear the disease, but if you make the virus replication defective, then it's actually a very beneficial property for a gene therapy vector. So as a first step, we did some work looking at keratinocytes and fibroblasts, and we're able to show that this vector infected both dystrophic EB keratinocytes as well as fibroblasts, and it did it quite efficiently. The next slide. And then we did some preclinical studies on mice, which looked very promising. And then this led up to a early phase 1/2 trial. And this is exactly how we put the medicine on the wounds. The medicine can't penetrate through tight junctions, so it can't penetrate through intact skin, but it does a very nice job getting into wounds where it infects fibroblasts in the wound bed, as well as the reepithelializing keratinocytes as they come in to heal the wound. And so we apply this once a week until the wounds heal. Have the next slide. And this is the results of the phase 1/2 trial, where the wounds on the top are the wounds that we treated. And as you can see, all the wounds here are healed that were treated with BVEC, except for one. And that one, actually, we re-enrolled the patient later and at a later stage in the trial, and then it healed. And so basically all the wounds showed healing. In contrast, the placebo wounds would not close, or they would open and close. And this is kind of the natural history of the EB wounds. If you look at this blue line here, this is like the placebo wound surface area. It goes up and down, it heals, then it gets bumped, then it reopens, then it heals, and it gets bumped, then it reopens. Whereas the BVAC, or the treated wounds, actually heal, and then they stay healed. You can see that for 3 months, it stays healed. And that's what collagen 7 does. When it gets into the skin, it makes the skin stronger, and it makes it resistant to getting blisters. And so there's two different ways you look at wound healing. One is time to closure, and one is the durability after the wound closes, that it stays closed. And this is what the collagen 7 gene replacement does, is it keeps the wounds staying closed. Next slide, please. And then we demonstrated the molecular correction. This green line here is the NC2 domain of collagen 7. The red is the NC1. And sometimes when we show these types of pictures, you can imagine maybe we're showing you the best image out of all the ones we took. But just to kind of show you a little bit more, we actually pieced together multiple low-power images all the way across the sample. And you could see really fairly consistent collagen 7 expression all the way across the sample. So this is a very robust molecular correction. Next slide, please. And then this just shows the anchoring fibrils. These are these little structures that connect the basement membrane onto the dermis, which is what collagen 7 is the component of. And as you can see here, these banded structures that are coming out perpendicularly from this central lamina densa and looping around collagen fibrils. This is, the gene therapy expressed and restored these really beautiful anchoring fibrils. So we see a very nice ultrastructural localization of the therapeutic gene product. And the assembly, it's basically, once you get collagen 7 in the skin, it's going to just assemble itself into the basement membrane. Could I have the next slide, please? And the successful phase 1/2 trials led to a phase 3 trial that we concluded. Next slide, please. And in this trial, we took primary wounds. This is a double-blind, randomized, placebo-controlled study over 3 sites for children and adults. And we took 2 wounds. One was treated with BVAC, another one was treated with placebo. And they were matched up for location, size, chronicity. And we also treated secondary wounds by open label just to give the patients additional benefit and to aid in the recruiting. And then we're currently in the open label extension period right now. And so, can I have the next slide, please? And so the endpoints are 100% wound healing at 6 months. And the secondary was 100% wound healing at 3 months. So this 100% wound healing is a very high bar. If it's like 95% healed, it's not good enough. It has to be 100% healed. And even with that high bar, we're able to show a significant proportion of wounds, statistically significant, that achieved that endpoint compared to placebo wounds. Have the next slide, please. And I think one of the most remarkable things about this, the efficacy is really great, but the safety is just really incredible. The field of in vivo gene therapy has never seen a redosable type of a product like this. So it's like a first in a redosable gene therapy, a first in a topical gene therapy, a first gene therapy for EB that's approved. It was the lack of significant adverse events is really a plus here. And so it's something that the patients really tolerate nicely. The next slide, please. One thing that we do see, and we are continuing to observe, and I think it's something that's very important for us to continue to look at, is the development of immune reactions against either the vector or the therapeutic gene product. About 50% of the population, at least in the United States, is HSV1 positive antibodies. And that's about what we saw in the trial. And some patients actually converted during the trial as well. But if you look at the patients who were positive for HSV1 antibodies or were negative, you didn't see a significant difference in the efficacy. Similarly, there are some patients that are actually some dystrophic EB patients that have collagen 7 antibodies. And this is, we don't know exactly why they have these antibodies. One hypothesis is that topical gentamicin is a fairly popular antibiotic, and it can potentially give read-through of some of full-length collagen 7, perhaps enough to be able to immunize the patient. We don't know, but we just know that maybe 10% of treatment-naive collagen 7 dystrophic patients have these antibodies. And then we did see some conversion of the collagen 7 antibodies during the trial. But if you look at collagen 7 negative antibodies versus positive, there's not a significant difference in the efficacy. So I think it's something that's important to study. I mean, before, we were just trying to get expression of our therapeutic gene product in the skin. Now that we have expression, we're confronted with new issues that we have to look at. And I would predict that it doesn't matter what kind of gene therapy you're using. You have to be looking at the therapeutic gene product, the antibodies that are developing against it. We have similar issues that we deal with, like with the biologics in psoriasis. Sometimes patients can develop antibodies against their biologics, make it so they can't use the biologics. They have to go on to a different biologic, which that's easy, but you can't go on to a different collagen 7. You have to stick with the collagen 7 that you have. So this is something I think that's going to be very important to look at for the future of our field, is the antibody production. And there are ways to be able to decrease antibodies, like, for example, IV IgG, and there's other ways to be able to tolerize the patients to be able to receive the gene therapy better. So, it's been a long history here in EB. EB was first discovered in 1875, and finally we got to the point now where we have the very first topical gene therapy. It was approved in the United States last month, May 19th, and I'm looking at in August to the patients are going to finally start to receive the therapy. I'm really looking forward to trying to get as many dystrophic patients in the United States on the drug as I can. And I really would love to be able to see the drug get out to all over the world to help patients. It's a very simple drug. It doesn't require a lot of technological facilities. Have the next slide, please. So in the United States, it's going to be home use. And then we're going to be starting to treat mucosa, like the mouth, the eyes, the esophagus, rectal area. Of course, the hands are going to be very important to address as well. And then the next slide, please. And I'd just like to thank everybody who participated in this trial. This was definitely a group effort with a lot of really talented people. It's been a great partnership with Crystal Biotech every step of the way. And especially I want to be able to thank all of the EB patients who donated their time. They're very altruistic in wanting to not only help themselves, but want to help the greater EB community. So with that, I'll pause and I can answer any questions if you like.